Scientists create mice from two dads after making eggs from skin cells. Scientists have created mice with two biologically male parents. The mice shown here represent a significant milestone in reproductive biology. CNN. Scientists have created mice with two biologically male parents for the first time, a significant milestone in reproductive biology. The team, led by Katsuhiko Hayashi, a professor of genome biology at Osaka University in Japan, generated eggs from the skin cells of male mice that, when implanted in female mice, went on to produce healthy pups, according to research published March 15 in the peer-reviewed journal Nature. The proof of concept research, the culmination of years of painstaking lab work, could expand the possibilities for future fertility treatment, including for same sex couples, and perhaps help prevent the extinction of endangered animals. However, scientists warned there's still much to learn before cultured cells can be used to make human eggs in the lab dish. It is expected that application into humans takes a long time, maybe 10 years or more. Even if it is applied, we never know whether the eggs are safe enough to produce a baby, Hayashi said. The skin cells reprogrammed from mice tails. The researchers took skin cells from the tails of fully grown male lab mice, which, as in male humans, contain one X and one Y chromosome, and turned them into induced pluripotent stem cells, or iPSCs, a type of cell that scientists have reprogrammed into an embryonic state. This process of genetic engineering, which introduces specific genes to create cells that mimic embryonic stem cells, was pioneered by Nobel Prize winning scientist Shinya Yamanaka. Induced pluripotent stem cells, which can be developed into any kinds of human cells, are widely used in biological research to model and investigate human diseases and develop the drugs. When the iPSCs are cultured in the lab, a few spontaneously lose the Y chromosome, which isn't essential for the growth of this particular type of cell, generating XO cells, Hayashi explained. The technology could help expand the future possibilities for the fertility treatment. The researchers cultured the XO cells and found that some cells developed two X chromosomes as a result of cells division making them chromosomally female. Treating the XO cells with the compound called the reversing increased the number of XX cells, the researchers found. From there, the team converted the XX cells into primordial GOM cells, the precursors of eggs and sperm, that are subsequently programmed with the signals to turn them into egg cells. Once fertilized with the sperm and implanted into a mice utellus, the eggs generated a living offspring. This story is particularly neat because it takes advantage of errors that are known to occur during culture of XY cells, which lead to loss of the Y chromosome and the subsequent gain of the second X chromosome, resulting in XX cells that are capable of generating living offspring, said Rod Mitchell, a professor of developmental endocrinology at the MRC Centre for Reproductive Health at the University of Edinburgh in Scotland. In a statement, He wasn't involved in the research. However, its possible application for humans, for example, for same-sex couples, remains to be seen. In the mouse study, very few of the embryos generated using mouse cells resulted in living live offspring, and the final steps required to convert the gum cells into eggs have not been reliably reproduced using human cells, added Mitchell, who is also a consultant pediatric endocrinologist at the Royal Hospital for Children and Young People in Edinburgh. Only 7 out of the 630 implanted mice only 7 out of 630 implanted mouse embryos gave rise to mouse pups. Hayashi said this low success rate, around 1%, wasn't down to the process of sex chromosome conversion, but the reality that cells cultured in the lab are typically inferior to those in the living animal. This is due to the optimal condition of the culture system, especially if the culture period is long, in this case 5 to 6 weeks, then the cell potential is compromised, Hayashi said via email. What's next? Hayashi's research has raised the possibility that someday same-sex couples may be able to have a baby who shared both the parent genes. It will be difficult to produce babies from the male to male human couples because of the both technical and ethnical, ethical reasons, Hayashi said, but it is theoretically possible to produce babies from the male to male couples, as shown in this study. He said that it would be more challenging to accomplish the reverse, that is, making sperm from female cells because they contain no Y chromosome, which is essential for making sperm. Duplicating an X chromosome, which male cells already have, is easier than conjuring up a Y chromosome in female cells, Hayashi explained. Glenn Cohen, a James A. Atwood, and Leslie Williams, professor of law at Harvard Law School, said the work raised thorny ethical and legal questions that the society needed to start thinking about. The technology could help the northern white rhino from going extinct. Nazim for foreground 30 and the daughter Fatu 19 are lost two of their kind on the planet. These issues include the embryo farming, producing hundreds of embryos to pick the best one, and the unauthorized use of person's cells. What happens to all the embryos created but not used? Does it violate ethical norms of respect to create so many potential human lives, then knowing that the vast majority will be destroyed or indefinitely stored, said Cohen, who is also the faculty director of Harvard Law's Petrie Flom Center for Health Law Policy, Biotechnology, and Bioethics. It is the most extreme case. Imagine an individual using sloth skin cells left on the bathtub by Brad Pitt, for example, to drive a sperm or egg in order to reproduce, he added. Saving animals from extinction. The technique holds a promise for conserving endangered species, although it is not known whether the process in the mice that resulted in the spontaneous loss of the Y chromosome and the duplication of the X chromosome would occur in other mammal species, said Mike McGrew, the personal chair of avian reproductive technologies at the Roslin Institute at the University of Edinburgh. This is very exciting finding for species conservation, he said via email. You could imagine that the many biobanks that are being established to capture genetic diversity stored for endangered species of animals by chance, so only or predominantly male cells may be conserved for some species. The techniques developed by Hayashi could help the northern right rhino breeding program, said Thomas Hildebrand, professor at the chair of wildlife, wildlife reproduction medicine at Puli University, Berlin, and the head of the reproduction management at the Leibniz Institute for Zoo and Wildlife Research. Only two animals of the species remain in the world, and both are female. Hildebrandt is attempting to artificially breed the animals with sperm and the tissue samples taken from now deceased male counterpart. It's a technology for tomorrow, but we have an option to create a genetically sound population. This is only possible with this stem cell approach, Hildebrandt said.